Hello everyone, I am Dr.sunali and today we are going to talk about classes of antigen, the T-dependent, the T-independent and the superantigens. Now this is a very important topic for your second year MBBS students as you can get lots of short notes for this. Same goes for your MD microbiology student and also for your competitive exams like your NEET PG, FMGE, INICET, you can get lots of MCQs from this topic. Okay, so let's decode the classes of antigens. First, we'll start with the T-dependent antigen. Now, your T-dependent antigen is an antigen which follows the protocol. It follows the rule. So, let's see what rules it follows. When the T-dependent antigen enters your body, it is processed by this antigen presenting cell. Okay, this antigen presenting cell processes it and क्या करता है फिर ये उसे present करता है on a plate. This is the plate. This plate is called MHC, major histocompatibility complex, okay, to the MHC, and in this case this is MHC2, to this MHC2 plate to T cell. This is the T cell. So your T dependent antigen enters the body. It is processed by the antigen presenting cell presented on a plate which is the MHC2 plate to your T cell. Okay. Now your T cell, after seeing this, it gets activated. Your activated T cell releases cytokines. Now these cytokines are specific cytokines. What will these specific cytokines do? It will go and stimulate your B cells to do what? To produce antibodies. Okay. This B cell is producing antibodies. Now you see these antibodies are like your sniper rifle. Okay. One clean shot, antibody dead. They are precise. This antibody production, because it's specific, it's being produced because of specific cytokine release. So it's precise and it's effective. Clear? Okay. So your T dependent antigen, this is your antigen presenting cell and this is the antigen. Your antigen presenting cell is presenting the antigen along with MHC2 major histocompatibility complex to किसको present कर रहा है? To T cell. Okay, this is the T cell receptor. Okay, now there is activation of T cell. Okay, and it releases specific cytokines. This specific cytokine will do what? It will go and stimulate B cell to do what? To produce antibodies. Clear? Easy? This is your T-dependent antigen. Now, let's meet the T-independent antigen. Now, not all antigens play by the rules, okay? Some don't like to follow protocols. Now, who are they? They can be your flagella, okay? Or they can be your capsule, bacterial capsule, or they can be your lipopolysaccharide. So, ये करते क्या है? What do? They directly bind to the B cell surface immunoglobulin receptor. There is no processing and presentation by the antigen presenting cell and no T cell required. That's why the name T-independent antigen. But how? Let's see. What happens is your T-independent antigen directly goes to your naive B cell. ठीक है? ये directly जाता है naive B cell के पास. उसके बाद क्या होगा? After that, this naive B cell has surface immunoglobulin. Which one? IgM and IgD. Okay? This naive B cell. Now, this T-independent antigen, what it does is, it is cross-linking to this B cell receptor at once, to many B cell receptors at once. And this cross-linking is non-specific. Okay? This is, you see, ये क्या है? This is the T-independent antigen. Okay? क्या कर रहा है ये? It is cross-linking to many B cell receptors at once. Okay? So what will happen because of this? Because of this, there is many B cell, there is many clones of B cell getting activated simultaneously. What is it called? It is called polyclonal B cell activation. What will polyclonal B cell activation lead to? hypergammaglobulinemia. Okay, hypergammaglobulinemia, that is non-specific antibody production, sky high level of antibodies, but it's not targeted. Okay, it's like spraying bullets everywhere without actually hitting something. Clear? This was your T independent antigen. We'll see here again. This is your B cell, and this is the B cell surface as immunoglobulin IgM and IgD. Okay. Now, what is this T independent antigen doing? It's coming and directly binding to your B cell receptor. Now you see, this antigen is binding to many B cell receptors simultaneously. So it will do what? There is polyclonal B cell activation leading to antibody production. Okay, hypergammaglobulinemia. Now, one point here, the antibody which is predominantly produced here is which one? It's IgM. Why? See, what happens is, early in immune response, IgM is the first antibody, the predominant antibody to be produced. But after that, क्या होता है? Class switchover happens, means IgM is changed to IgG, IgA. It depends on the cytokines produced by the T cell. But here, no T cell, no cytokines, B cell is the only player. So, there will be no class switchover. The only predominant antibody which is there is IgM. Clear? This was your T independent antigen. Now let's see the difference. So for your T dependent antigen, the structure of the antigen is a complex protein. Okay, it's more commonly encountered. It's structurally complicated. T independent, these structures like your polysaccharide, flagella, capsule are simple structure. They are not complex. Antigen processing for your T dependent, it's needed, must be processed by antigen presenting cells and presented to the T cell. T independent, not needed. Directly bind to B cell receptors. Okay. T cell help for T dependent, required. T independent, as the name suggests, not required. Memory, yes, it's present for T dependent. T independent, absent. Okay. Class switchover, what did I tell you? For T dependent, will it happen? Yes. From IgM, it will change to IgG, IgA, IgE, depending on the cytokines produced by the T helper cell. But for T independent, the cytokines are not being produced because no T helper cell is being stimulated. So, mostly IgM and very rarely, very sometimes IgG type 3. But remember IgM. Okay. Type of B cell activation is monoclonal. Why? Because specific B cell is getting activated due to specific cytokine release. In T independent, it's polyclonal. Multiple B cells activated polyclonally. I showed you, right? Because that antigen was cross-linking to many B cell receptor at once. So, non-specific activation. Clear? Metabolism, it is T-dependent antigen is rapidly metabolized. T-independent is slowly metabolized. Okay? Antibody response, obviously T-dependent has a strong, diverse response with all classes produced. T-independent, restricted, weak, mainly IgM. Okay? This was your T-dependent and T-independent antigen. Now, let's move ahead. Our super antigens, our supervillains. Okay? Now, there is another group of antigen. These antigen, they are like supervillains. Okay? Normally, what happens? Your antigen in a normal scenario for your T-dependent antigen, your antigen enters, processed by this antigen presenting cell, presented on this MHC plate to your T cell. This is a very normal scenario, but your super antigens are what? Supervillains. They don't like to play by the rules. They like hacking the system. So ये क्या करता है? What they do, it hacks the system. How? It bridges the MHC2 of your antigen presenting cell and the variable beta region of your T cell receptor. It bridges them together. So what is it doing? It is hacking the system. Okay, it attaches itself to both the MHC2 of antigen presenting cell and the variable beta region of your T cell receptor. It will lead to what? It will lead to chaos. Okay, this is not something normal for them, for your cells. So what happens to your T cell? T cell gets stimulated non-specifically. What does it lead to? It leads to chemical chaos. There is miscommunication. The T cell getting activated non-specifically, starts blasting cytokines like it's holy. It's not a good kind of holy. This causes destruction. This kind of blasting of cytokines is known as cytokine storm. It causes destruction, it causes death. And this cytokine storm further stimulates your B cells polyclonally, okay? And it leads to hypergammaglobulinemia. Now, this part is similar to your T independent antigen. Same thing was happening. B cell, because of non-specific stimuli, it is getting stimulated polyclonally. Many B cell clone activated at once, leading to hypergamma globulinemia. Okay, non-specific antibodies are being produced. Clear? This is your super antigens. So, normally what happens? Antigen presenting cell presents antigen along with MHC2 to T cell receptor. This is your rule follower, the T dependent antigen. But your supervillain, your super antigen, what will it do? It will bridge this MHC2 on the antigen presenting cell and the variable beta region of this T cell receptor. It bridges them, hacks the system. Clear? Causing cytokine storm, T cell exhaustion and death. Clear? Now, this cytokine storm leads to various diseases. Let's see. And what are the organisms which produce cytokine storm? We'll see which are the super antigens. Okay, so we'll first start with the bacterial super antigens. In bacterial super antigens, first we have Staphylococcus. Your Staphylococcus produces toxic shock TSST, which is a super antigen. Exfoliative toxin is a super antigen, and enterotoxin is a super antigen. What will it cause? It will cause cytokine storm. Okay. Now you see in Staphylococcus, these are the super antigens. So here is your mnemonic. In Staphylococcus, you see what are the super antigen? Toxic shock, exfoliative toxin, and enterotoxin. Clear? Now comes to you Streptococcus. For Streptococcus is streptococcal pyrogenic exotoxin. It speaks AC, SPEAC, streptococcal pyrogenic exotoxin AC. Okay. So your mnemonic here is it speaks AC. SPE is the streptococcal pyrogenic exotoxin, A and C. Next is your Mycoplasma arthritis mitogen, Yersinia enterocolytica and Yersinia pseudotuberculosis. Okay, this is my favorite. Okay, it's my Mycoplasma, Yersinia and Yersinia. Okay, remember it like my. So these are the bacterial super antigens causing cytokine storm. Let's see the viral super antigens. Okay, in viral super antigens we have Epstein-Barr virus, cytomegalovirus, rabies nucleocapsid and HIV, the NEF gene product. Now, how to remember all these? You can remember by this mnemonic. Every crazy rabbit has HIV. E for Epstein-Barr virus, C for cytomegalovirus, R for rabies, H for HIV. Okay. Now, next is your fungal. Fungal super antigen is Malassezia furfur. Okay. This is F for fungal, F for furfur. So, Malassezia furfur. This is a list of very important list which you should remember because this comes as MCQs. Okay. Now, what was the disease the super antigen cause? This for Staphylococcus, the TSST1 toxin, that is the toxic shock toxin. What disease it will cause? It will cause toxic shock syndrome. Clear? The exfoliative toxin, what disease it will cause? It will cause scalded skin syndrome. Clear? Enterotoxin will cause what? It will cause food poisoning, right? It will cause food poisoning. Similar to it, we have other diseases also which can happen due to your super antigens like your atopic dermatitis, Kawasaki syndromes and many more. But this is the list which you should remember. Okay. Thank you. Now that's your classes of classes of antigens. Now if you found this helpful, hit the like and subscribe button and share it with your peers. Thank you.